Thank you very much for this very kind introduction. I can make it only worse from here. So, basically, before we talk about data, I would like to show you a little bit of the background. Why we actually have so many difficulties to understand inflammation in general and what inflammation is doing in the brain. So if you look at inflammatory mechanisms, let's imagine that we cut our finger and then it's immediately releasing this wound. A lot of mediators, very simple mediators, which aim to recruit inflammatory cells, the capillaries will dilate, and locally there will be a coordinated response in order to heal this wound and to fight infection, because every single event of injury is a potential risk for infection. And during evolution, being infected is the top one danger you want to avoid. So you overreact and you make sure that there's no infection. Okay? So what happens that these inflammatory cells go there, and then you can see those typical signs that your finger turns red, it swells, it's painful, it's warm, and sometimes you cannot use it for a week or two, and if there is chronic inflammation, you can see how painful it is. Now, this thing usually goes away and it heals and everything goes back to normal. And if you look at our peripheral organs, let's imagine now we are not talking about our fingers, but everything which is beyond the central nervous system, the brain and the spinal cord, that most of the blood vessels are kind of leaky a little bit. It doesn't mean that the blood would leak out, but a lot of proteins and cells can run into the tissue from the capillaries. There are lymphatic vessels which are even more permeable. So there is a very intense communication. If you eat a drug, that will show up in all of your organs. This is how medications work. And you have a lot of specialized immune cells and also mostly a high level of regeneration, although chronic inflammation can cause harm, as we know from many diseases. In the brain, your nerve cells have high level of activity, so the brain would consume approximately 10 times more energy than its weight compared to other organs of the body. So you need an extremely dense capillary network, and you need to regulate very well blood flow, because if it stops for a few minutes, brain cells would die and it's gone. So you need to restrict also blood proteins to leak into freely to brain tissue. You have blood-brain barrier, and you also have a limited access to peripheral immune cells because it's very, very dangerous to cause inflammation in the brain. And you have a very specialized immune cell, which you allow during early development, already during the first trimester. In people, it's 4th to 5th gestational week. And these cells would go into the brain and carry whatever they carry and, to the developing certain nervous system, and then they will be a self-renewing population throughout life. So inflammation is not only linked functionally to many of the brain diseases we know, and almost all brain disorders have an inflammatory component, and the field and multiple fields now tend to agree that it's important. But if you look at what type of cells coordinate inflammatory responses in the brain, and what types of cells would protect the central nervous system against anything else which comes from outside, then you would see these microglial cells, which were noticed by very famous people, including Alzheimer himself, more than 100 years ago, in postmortem brain tissues, in patients with brain injury. They found that these cells change their morphology, they gather around neurons, they appear to kind of eat up debris. Don't forget, at this time, it was also a matter of debate whether neurons are individual cells or just interlinked somehow. So there was no modern physiology to talk about it. Now, if you look at these microglial cells, they have these very motor processes. They keep moving. Sometimes they touch blood vessels. So I just go back and you can see it again, this video. And also, you can have microglial processes interacting with neurons. So this is a living mouse brain. In red, you can see the neurons, and in green, you can see these microglial cells. And these cells just survey their microenvironment, and that's what they do. If there is injury, they can very quickly react. So you can see here, injection of ATP, a danger molecule, into the brain. And within a few minutes, these microglial processes go there and isolate the area. So ATP is inside the cells. We have a lot of it. If it leaks out and the extracellular ATP concentrations increase, that's a danger signal. So the brain uses this system very wisely, and other cells, of course, in the body use this system very wisely to report danger to these immune cells. Now, another thing, if there is a blood-brain barrier injury, you remember I told you that we isolate the brain by having a barrier provided by endothelial cells and also astrocytes and other cells. If there is stroke or something like an acute injury, you have blood-brain barrier injury, and you can see the leakage of this dye into the brain parenchyma. It's even worse that these microglial guys are there and know exactly that something is going to happen half an hour earlier. And there is data that these cells can also protect this tissue somehow. Now, if there is infection indeed, what would be the function of an immune cell? You can see here a viral infected neuron in the cerebral cortex of the mouse, and I'll show you once again because it probably makes sense to see the video. And these microglial cells gather around and would isolate this neuron and rip it into pieces within a few hours. So this happens in the human brain as well, and if you identify one single phagocytic microglia, which is indicated by the debris of the neuron inside the cell, that indicates that 10 times more phagocytic events would take place. So you just don't see it. But these barriers are formed, and remember this barrier because I will show you something very exciting on the last slide of the talk about this. Another very important thing is that these microglial cells change their phenotype as there is inflammation in the periphery as we get older, or if we have chronic inflammation, for example. So this is a study we've done with clinical colleagues in Manchester. These are patients at risk of stroke in the upper row, and with multiple risk factors of stroke and chronically elevated C-reactive protein, which is a marker of inflammation. And in the bottom row, there are patients with low number of risk factors and low CRP. And this colorful story here is indicative of microglial activity. This is TSPO, and positional emission tomography, showing how active these microglial cells in these patients who are neurologically negative. They had nothing, no symptoms whatsoever. 2 of them died within a year in stroke. And if we model this in animals which have chronic inflammation, these corpulent rats, they weigh approximately 1 kilo. They don't move too much, but they have chronic inflammation, metabolic disease, arthritis, so kind of elderly patient who has multiple issues and inflammation. You can see that in their brains, microglial activity with another ligand, but this is also positive emission tomography, increases compared to the lean animals, but also over time as they get older. So we not just have a microglial cell which is supposed to supply all the functions of immune cells in the brain, but this function changes with inflammation or with aging, meaning that these cells wouldn't be able to fulfill their function. Now, where these cells are, let's have a look at the brain. In green, you can see the microglial cells. They are pretty much everywhere. And in magenta, you can see cortical neurons. If you zoom in a little bit, you can see that the microglial cells are kind of distributed. Sometimes they touch the neurons. And if you just look at where they connect to the neurons, you can see that not just touch the neuronal dendrites, but also the somatic compartment of the neuron, many places. Almost all cells in the brain receive these microglial processes, and don't forget that these are multi-processes, though everything is moving except for the neurons, and kind of checking these connections. Now, I try to show you it again, so you can see that these processes not only contact these hotspots on the neurons, but they also recontact these sites. So they actually, and this was a big question when we started to do this research, would there be hotspots on the neuronal membranes which microglia would recognize? Would neurons tell something to these microglial cells? Or this is just a random scanning event, as the field thought that they would just move their processes everywhere in the tissue. And it turned out that actually there are hotspots. And I just don't want to tell you the whole story because I think this would be very complicated. But the take-home message is, what you can see here is end of a very long anatomical research program. So at this stage, we don't talk about function. And this is now electron tomography. So what you do, you have your in vivo imaging. You see how the system works. You have your anatomy. You you have these very nice fluorescent pictures showing microglial neurons, and then you zoom in, you process the samples for electron microscopy, you label with immunogold particles those microglial receptors which you know to be important for talking to the neurons, which you can see in yellow here. And you can then reconstruct these very, very small connectors, which are just a few tens of nanometers in size. And this would determine, because there is a lot of them, microglial cells would touch a neuron on the cell body 2 to 3 times an hour, which is 500,000 connections between a single microglial process and a neuron over 10 years of life. So try to imagine how many chances these cells would have to destroy or to protect these neurons if they function or malfunction. And just to make it short, we have a structure with mitochondria, which would be kind of the energy generators of the cells, also very important for a number of diseases. So microglial activity is one of the earliest things which goes wrong in neurological disorders, together with blood flow and increases in microglial activity, even before neurodegeneration takes place. And you can see that this system has a very, very specific anatomical feature, because if you look at yellow and in cyan, you can see 2 different receptors through which microglial neurons could communicate at these sites. And in electron microscopy, you can see that this is a neuronal intrinsic feature to have their own receptors, and then microglial kind of hands on them would be limited or restricted to these areas where these neuronal receptors, Kv2.5 channels in this case, cluster. So can we tell whether it's functional? So then we have to take these brain cells out and do in vitro studies. So what you can see here is a microglial cell in red, but then we use an alien, we use hex cells, which are not in the brain, but they fortunately don't have these channels. So we can transfect them. We can ask them to express something through which the neurons normally communicate with the microglia, and we can see whether the communication takes place. You can see that these small clusters of these green channels communicate with the microglial processes. If we make it wrong, if we knock out a small piece of the peptide so that these neuronal channels cannot cluster, microglia can no longer communicate with them. And you lose this effect. So you prove that actually at this level, the communication indeed takes place between these 2 molecules. Now another thing, would this danger signal be released from neurons even during their normal activity? That was another surprising thing. Because in the cell, you have a lot of ATP. The cell uses it as an energy currency. But the cells also use ATP much, much lower levels, 100, 1,000 times lower levels for communication. They leak a little bit, and they tell their microglial cells, I'm okay. If this communication is interrupted, microglial cells know exactly that something is going wrong. Here we depolarize neurons, so we cause activity, and we can show that ATP is released. And then another story comes, how can we tell whether it's functional? Whether there would be interaction between microglial cells and neuronal fate. Would these cells be important for neurons, or they just touch them and communicate to them, and then nothing really important happens? So we managed to get hold of a tool with which we can eliminate microglia from the brain. This was designed as a tool against synovial tumors in the knee, where macrophages upregulate this, and researchers noticed that actually the microglia go away from a normal brain if you feed this drug to mice. And I got in touch with this company, got the drug in, and it was surprising. You can have an animal feeding this drug to the animal, and the microglial cells disappear completely with apoptosis, so you are not inducing inflammation in the brain. And what happens, then these microglial cells seemingly have no role. The animals are happy, they survive. The young animals can be without microglia for 2 months, even they learn a bit faster. And he said, come on, I mean, we have been working on these blood cells for a long time. You take out them and nothing happens. Okay, let's cause injury. And I don't make it very long, but when you cause injury and you see that these microglial cells can communicate with now the injured neurons, in this case we cause experimental stroke. We occlude the blood vessel, we have an area in the brain where neurons will die, but around them neurons are very susceptible to injury. And then many of them will die in several hours. And this will determine whether a patient will be able to move with their leg or their fingers anytime during their life. Because if it's gone, no way you can put it back. Now these neurons here fight for their life, and microglia are touching them with their hands. And when we take out microglia, you can see that these neurons start firing, start working, oscillating in a very weird fashion, which you never see when microglia are around. And they die much quicker. And what you see that intracellular calcium, which neurons use for their signaling, goes up. And what happens also is that these neurons will die very quickly in the infarct, showing that microglia are functionally important, and as a result, you have a larger brain injury. These animals do functionally worse, and you can show it with different approaches and tools. I'm not going into detail, but you can block now these same receptors, the P2Y12 receptors we identify to be important for the microglia neuron communication, and you can have the same effect. Larger injury, more neurons dying. That was certainly not welcomed by a lot of people who wanted to claim that microglia are evil guys in the brain, so we had a lot of fight with that. But I show you some other things which we had also a lot of trouble with to publish. Some of these data were actually stolen also during the publication process, which is not necessarily a nature of science, and this is kind of a random thing, but it took so long that we had very helpful people who didn't like to see what we are doing, which is also sometimes something, it's not part of science, but also can happen if there is a big debate about something. So don't take it as an operation for science. But these cells also communicate, microglial cells, with blood vessels. And I showed you that with the little fingers, they can touch the blood vessels and work around. So when you look at single microglial cells and reconstruct them in 3D, you can see that at the same time, they have their processes on multiple neurons and multiple blood vessels at the same time. And you know that in the brain, one of the most important things is to drive blood to the area of activity. So what the brain is doing, if we need neuronal activity, if we need neurons to fire, it's going to consume a lot of glucose and blood. So you direct blood exactly there where the neurons are active. This is what functional MR, for example, is using. And the idea was, okay, could this process be influenced by microglia? So we reconstructed in the mouse brain and also in the human brain them in 3D from the large vessels which penetrate from around the brain and they dig deep, and then everything, all the blood which gets into the brain would derive from these vessels. And microglia are everywhere. And just again, to make it very short, these microglial cells communicate with all the cells which are important to regulate blood flow. And they have their receptors directly on the surface, and exactly they communicate with the mitochondria, similarly to that seen in neurons. So if we use activation of neurons, if we trigger neuronal activity in the somatosensory cortex by tickling the whiskers of the mouse, you probably remember there was a presentation about it. You can see with laser spectral contrast imaging, this is a method with which we can look at neuronal activity indicative of blood flow in the brain. So we can get blood flow, but behind that certainly there is activity-dependent coupling, as we call, between neuronal activity and blood flow. You can see in the mouse brain that wherever we excite the whiskers on the other side in the brain, there is going to be increased blood flow. So would microglia be important for that? Yes, if we block them or we deplete them, this response is impaired. So how can we prove that it's really microglia in real time? So we developed a mouse in which we can chemogenetically target these microglial cells. So we can inject the drug intraperitoneally, and none of the other cells would respond, only microglia. So when I put the animal under 2-photon microscopy and I kind of fire the cannon, these microglial cells would depolarize, and I can tell exactly what they are doing with the neurons in the brain, either with or without injury. So you can see here a microglial cell isolated here. And whenever we hit them with this chemogenetic compound, which is not recognized by anything else, all the cells just express the receptors and none of the other cells in the body. And you can see that microglia stop the processes. But another very exciting thing happens. They become insensitive to ATP. So you remember this danger signal. They can no longer respond to that. So basically, we have a tool to make them insensitive and see these kind of disabled microglia, how they would react. And they react exactly the same way. We have a deficient coupling, a deficient blood flow in response to somatosensory stimulation. And another model, very quickly, just to how we do vasodilation. So in the brain, there is very little protein in the cerebrospinal fluid. So there is not a single site for sensing low oxygen or high carbon dioxide levels. And in this case, there is a mechanism to vasodilate through high carbon dioxide, which is called hypercapnia-induced vasodilation. And many brain areas can do this. And what happens is if you have high CO2, pH would go down, and this pH drop would increase, basically, vasodilation. So you can see microglia respond very quickly. And if we take them away, this vasodilation is impaired. And you can see that in a mouse in which microglia are dysfunctional, these blood vessels cannot properly dilate. So these things also take place in disease. And as I said, in the early phases of neurological disorders, you also have decreased blood flow and decreased coupling response. So here, the patient has no neurodegeneration, but no longer would give a proper response to stimulation. And if other things happen, this brain would be fragile. And just to finish off, the last thing I would like to show you, this is unpublished data, but the paper is going out soon. So we have a large collection of brains from people who died in COVID-19. And we mapped the microglial function in there. We used all the markers and all the tools to tell what's wrong with them. And there are inflamed vessels in these patients in multiple brain regions. This is why the symptoms are so diverse. This is not localized to a brain region like Parkinson's disease. You have a brain region, substantia nigra, where the neurons would be dying, and that would have a stereotypical phenotype. We have a lot of symptoms because this brain doesn't know where inflammation will take place. If it will take place in your thalamus, you would have sensory symptoms or you wouldn't sleep properly. If you have inflammation in your cortical areas or in the hippocampus, there will be impairment in memory or coordination, or there will be an irritative behavior, psychiatric symptoms. But what you can see remarkably that these microglial cells not only change their activity, but they downregulate those receptors which I showed you to be important to control neuronal function. So in the COVID brains, and now we are looking at this in patients who are through an infectious period, and then they recovered, and they have still neurological symptoms. With other tools, what happens in their brains? Because there is a very strong evidence now emerging that the malfunction of the inflammatory system and the uncontrolled inflammation and the microglial dysfunction would be a very critical factor for neurological symptoms in COVID-19. So just to finish off, two kind of take-home messages. On the left-hand side, we have a brain, and this is a human brain. The lady was 102 years old when she died in a non-neurological condition. You can see the neurons in red, and you can see the microglial cells in green, and every single neuron is contacted by microglial cells through the connectors we discovered, which means that microglial cells would have had a lot of opportunities to eat up these neurons alive. We quantified this. If you put them in a dish, microglial cells would be able to eat up your whole brain within 24 hours. They don't do it for 100 years. So the regulation must be extremely important to understand. And on the other hand, I would like to kind of share with you my crazy idea about the grumpy maid model of microglia. So if you think about the Victorian household, and you probably read the books of Agatha Christie or Sir Arthur Conan Doyle, the Sherlock Holmes stories. Now, there is always a maid in the house, and the maid is an extremely important person. Nothing would function. The lord of the house would be sitting in a chair and would be able to do nothing without the maid. And certainly, if the maid doesn't provide tea to the landlord, or to the lord of the house, then there will be dysfunction. So we can cause injury, not just directly, we can cause injury by not allowing inherently protective or important processes to take place. And certainly there are occasions when learning these books, the maid would put some poison into the tea of the lord of the house, and that's a bad story then. So microglia might also poison sometimes our lives, but not because they are evil inherently, but because they are malfunctioning. So with this, I just want to thank for the hard work of the people in my lab, founders, and also for your attention. Thank you very much.
Thanks a lot. Thanks a lot, Ádám, for this very thought-provoking presentation. I wonder whether there are any questions from the audience. We encourage you to ask.
So thank you for this wonderful presentation. My question is, how did you take those pictures that we've seen in this presentation?
The fluorescent pictures, you mean? Yeah. So we have multiple microscopy technologies, I would say, which have been developed, and we are very lucky in the Institute of Experimental Medicine because this is one of the top neuroscience institutes. So people certainly developed and use these technologies for other purposes, to look at neurons only. And when I came back from Manchester after many years, my idea was exactly that. I either have no ideas and I have to come back where ideas are, or I have ideas and I have to come back to a place where the tools are there. And that was exactly the case. So we didn't have to develop, at least at the beginning, those tools, those microscopy tools, which a lot of energy, was spent on developing them and buying them for many other research groups in the institute. And I mean, in vivo two-photon microscopy is mine. I bought it from my ELC grant, but it was also custom built so that it allows us to stimulate neurons or to record. But there are many other core facility machines which are used by many researchers in the institute so that we can kind of get hold of the latest technology. So you are very welcome anytime to come to the institute and see whatever is there.
Thank you for your amazing presentation. I would ask that we could see in this presentation that the microglial cells are helping neurons and support them if it's needed. My question would be, could we use this knowledge to support neurons when, let's say, we have a patient who had a really hard stroke, or he has Parkinson? Could you find a cure with this kind of microglia effect?
Well, thank you. This is an excellent question, and exactly this is what we are trying to do. There are good news and bad news. The simple answer is yes, of course. That would be the aim. One, you could modulate microglia or change their activity, or even genetically modify them to kind of like Trojan horses carry things into the brain. One problem is they are there in the CNS in the first trimester. And this is, again, appears to be a problem for neurodevelopmental disorders, because if microglia are hit by a single inflammatory stimulus, they will carry the malfunction with them, and because they are having no supply from the blood, these cells would kind of keep this malfunction for the entire life. So many of the disorders, which are neurodevelopment disorders, autism is a typical example, would have an inflammatory component, but unfortunately, if the system is wired in a bad way, there is not much you can do. But the stroke is a good example, and neurodegeneration is a good example, because microglia are very sensitive sensors. So first, we could use them as a diagnostic tool to tell exactly very early on that something is going to happen. Second, we could genetically engineer, we could pharmacologically modify them. And there is a fourth possibility, which is extremely exciting. You can take out microglia from the brain without a problem. Unfortunately, the clinical approaches which try to restrict microglia failed because you cannot do this in patients with risk factors. I cannot tell you more because of confidential issues, but that happens. But if we could, you could even transplant microglia into the brain, and that's something a lot of people are working on.
Thank you very much. I wonder whether there are other questions from students. We very much encourage students to ask questions. Yes, back there, please. Ado. This is the fastest time.
Physical exercise is also very healthy for the brain.
Thank you. No need to hurry. Thank you for this presentation. But I have a question. You spoke about that microglia is capable of isolating infections. Is it true? And if it's true, can we modify this to create basically pockets in the brain where the infection is contained for the rest of our life?
Yes, that's another exciting issue. Microglia themselves wouldn't become infected. This was our first question when we saw these. That was kind of a neurotropic virus infection model. And the microglia have a very, very nice tool so that they don't become infected. But there's another problem. When they start eating up neurons, or when they see danger themselves, so that they see the viral proteins, they don't get infected, but they might die. And if they die, they can be replaced somehow by the proliferating microglial cells. But unfortunately, as we get older, the flexibility of the system is kind of running out. And this is very similar to the hematopoietic system, where in elderly people, the immune functions are not that good because you don't have the cells in the bone marrow to supply this system. And you always have less and less cells which are functioning properly and more inflammation and dysregulation. So we think also it happens with microglia, that if they encounter viruses, if they encounter debris to phagocytose, there will be more and more problems they should resolve, and then this makes them dysfunctional at times.
So thank you for your presentation. And my question is that you mentioned autism, and are microglials responsible for psychiatric disorders too? Or?
Yes. So that's another important thing. Let me take another example, not psychiatric disorders, let's take stroke as an example. If you have stroke, microglia will be very active on the side of the injury. But the remarkable thing is that they become also active on the other side of the brain. And if you look at patients, more than 1 year after stroke, these microglial cells are still active. Now, if you go back to psychiatric disorders, this is kind of the problem with inflammation in the brain. You can trigger inflammation quickly, but these cells wouldn't be silenced that quickly. And most likely this is a problem with psychiatric disorders, that if there are other stimuli which we are not aware, like chronic inflammation, which keep microglia kind of either too active or malfunctioning, those already existing problems can be intensified or new problems come. Just one example I have to tell you. Psychiatrist colleague of mine with whom we had some work together in the COVID story tells me that patients who had been seen by them 10 years earlier for psychiatric conditions, anxiety or loss of memory or irritative behavior, whatever, they are getting their medication and they are fine. Then COVID comes and the same symptoms 10 years later come back. And it's very likely that inflammation has a very key role in that. And if it's true that microglial cells become active, it can bring up symptoms. Another example, very short, in kids, in children, psychiatrists see a lot of problems after COVID. Very, very weird symptoms, which could also be influenced by inflammation and microglia. And that's a big challenge, how to deal with this.
Thank you very much. I wonder whether there are other questions from students. If a short question, short answer, we might still have time. Professor Brakonso is very mad at me anyways. But so if there are none, then I have convinced Professor Deli to ask her question during the coffee break, because they have many opportunities to talk to each other. So thanks a lot again, Ádám, and congratulations.